when I ruled the world.
Okay, we're at 109. Co-host will continue to monitor the waiting room. I get to start this show. So what we're talking about today is the virus life cycle. There's a lot to learn. So I cut it down to the first two parts, which is viral entry and replication of the RNA. And these pictures are in my slideshow, so you'll learn more about them as we go along. As always, we'll have some places at which I'll ask for questions, and you can put your question in the text chat, or you could raise your hand and ask your question out loud. Here we go. So this is a complicated slide. You don't have to understand it, but I used it because it shows there are multiple stages to the viral replication process. This is the virus itself up here. Attachment and entry is step one. Then it gets inside the cell, and then this is all RNA here. And I'm calling that step two. Step three is over here. I'm not going to talk about that today. Step four is the virus comes back out, but it's multiplied. So one virus goes in and maybe 1,000 or 10,000 come out. And so we're focusing on this half of the diagram today. This is a pretty good representation of the virus structure. So it has an inside, and inside is where the RNA exists. So some viruses are DNA viruses, some are RNA viruses. This coronavirus is an RNA virus. And then the outside is dominated by these spike proteins, is what they're called, spike proteins. And these are what the virus uses to attach to a human cell. So we'll come back to this slide. I have it duplicated later in, but that gives you the basic structure of the virus. Step one, virus binds to cell receptor. So here's a cartoon, human cell target. Here's a cartoon of the virus that has these spike proteins on the outside. And infection requires the virus to bind to a specific target on a human cell. So that specific target is called a receptor. If there is no receptor, the virus will pass this cell by. It's like Passover. If this does not exist on the particular cell, then the virus goes floating by. I have a 15-second video clip here which illustrates what happens when it does attach, what happens when it binds. This is from Cornell, as you can see. So this picture should look familiar to you. There's the virus with the RNA inside and the spike proteins outside. What's not well labeled here is this is the human cell. This is the human cell, and these Y-shaped things here are going to represent the receptor. This is only 15 seconds. I might play it twice. So the important, for the virus, the important part is to get this RNA inside the cell. And these proteins on the surface, their purpose is to, it's twofold actually, twofold purpose. One is to bind, to stick to the right cell. And secondly, to allow this viral membrane. This circle here is a viral membrane, and there's a membrane on the human cell, and they have to merge, or it's called fusion. They have to merge. So you'll see that when I run this a second time, there are three stages, right? So first the spike will bind. then this fusion occurs where the membranes join, and that allows the RNA to go inside the cell. That's the first stage in the infection, virus getting its RNA into the cell. So there is a specific protein on some human cells. Not all, but some human cells have this specific protein. It's abbreviated ACE2. If you're a scientist, you know what ACE stands for, angiotensin converting enzyme. It's not important. Actually, it is important, but it's not important for you. So we'll call it ACE2, and it's on the human cell. And it's supposed to here represent that it's the mechanism that allows the virus to bind to the cell. Boom, right there. This right-hand picture is the same, but a much more detailed perspective. This blue thing here is ACE2 on the surface of the human cell. This red is the viral spike protein, and you can see it reaches out and fits with the ACE2 receptor. And that's, once it's stuck, once this virus has stuck to the cell, then it will start the sequence of getting its RNA, which is not shown, but the RNA is in here, getting it into the cell. So this protein, ACE2, as the title of my slide says, is abundantly expressed by lung cells. So not every cell puts ACE2 on its surface. This picture probably doesn't mean much to you. And realistically, if I just had the picture, I don't know what it says. But the authors of the paper are pointing out, look at these dark spots here, which show the presence of this protein, ACE2. So where is ACE2? This is lung tissue, and this is also lung tissue. And the stain, the dark spots indicate ACE2 is here in these lung cells. ACE2 is here in these lung cells. And so this was published by scientists from the Netherlands. And in their abstract, I highlighted it. The most remarkable finding was the surface expression of ACE2 protein on lung alveolar epithelial cells. So there's a lot of it in the lung cells. And this is what allows the virus to specifically attack the lungs of humans. This, I love this picture. It taught me so much. So first you have to know, what am I looking at? These are human, I'll just say lung cells. Alveoli is the special part of your lung where the gas exchange takes place. If you just say human lung cells infected with the virus. So these were cells that were taken out of a human. You can grow them in vitro, means in a little dish. So they're growing in a petri dish. And then they infected with the virus. Viral particles are seen in double membrane vesicles. So these little dots here, which are numerous, those are the virus particles. And you can see they're accumulating within this human lung cell. And then they're saying, here's the virus that's come out of the cell. Look at them. So the virus replicates inside, but then in order for it to continue its life cycle, it has to come out. And so this is the virus that has been shed by the cells. All right. Let's see what my next slide is. Oh, question time. Okay, question time. So I'm hoping that you'll ask me a question about something that has already been in the show. And so I'll pause here and look for questions in the chat. Ray has a question about the song.
The answer is on the screen.
There we go. Okay, here's a question from Mariana Kerfoot. Do all lung cells have ACE2 receptors? Okay, excellent question. So in this slide, you can see they specify type 2 cells. And so there are different types of cells in the lung. Type 2 is the ones that particularly express ACE2. So type 2 goes with ACE2. That's a good way to remember it. This type of cell turns out to be extremely important. And the fact that it is attacked does a lot to explain why this virus is so deadly to humans. So that'll be in my next set of slides. All right, Luca Elganian. Why is the leaving its host cell part of the virus life cycle? Okay, so that's a good question there as well. The typical, if you're a virus, the typical way you proliferate is you say, I will infect this person, but I know that this person in a week or two is going to develop antibodies, develop immunity to me, the virus. So I can infect this person. I can probably live there for about a week, but after that, the human will kill me. So the virus life cycle is infect a human, multiply, get out of that human into another human who is not immune, live in that human for a week, get out of that human into another human. So for the virus, it's like a hotel stay. I stay here for a week and then I've got to check out and go to another hotel. So that's why if it never came out, then it would infect that one human, it would die, and maybe the human also dies, but that would be it. We infected one person and the epidemic is over. All right, Anna Chang has a good question because she read the fine print. What are lamellar bodies? Thank you, Anna. So these things that look like Van Gogh's Starry Night, I was wondering, what are these things? And it turns out that these are called lamellar bodies. Lamella means a layer, and you can see these multiple layers, like an onion. Ogres have layers. Ogres are lamellar, like an onion. Hold that thought. I think I'll repeat this slide. I think it's repeated, and I'll be able to talk about why are these so important? What do they do? All right, and Luke is now saying, how does the virus know that the human is going to develop antibodies? And of course, the virus doesn't, but viruses are a product of evolution. So you can imagine nature, through the process of random mutations, creates a virus, which is what happened with this one. Nature created random mutations and made this virus, the SARS-CoV-2 virus, the COVID-19 virus. Let's say nature made it so it could never get out. Then we wouldn't have it. It was a bad design of a virus. It went in, it died, game over. So the reason when we say the virus knows it has to get out, it doesn't. But the only way it can survive is by getting out. So evolution, survival of the fittest, right? Survival of the fittest. This virus gets out, it survives. All right, thank you for the questions. I'm going forward now. There'll be another chance to ask questions in a few slides. Okay, here we go. The type 2 cells. These are lung cells. I probably should have just said lung cell type 2, but the specific name is alveolar type 2 cell. In this diagram, you can see alveolar type 1 cell, alveolar type 2 cell. Most of the, this is the same picture twice, but they have different pluses and minuses. So I'll start with this one and then I'll go to the second one. The purpose of the lung cell is to enable gas exchange. So there's an air space. It has oxygen in it. Those oxygen molecules are supposed to go through the lung cell into the blood vessels, which are nearby. If that happens, you say, I'm breathing normally, I feel good. If that stops happening, then you say, I can't breathe. I'm trying to breathe, I can't breathe. The oxygen's not getting into my blood. So those are the people who have been sickened with this disease. Shortness of breath, they need to go on a ventilator, they get increased oxygen to try to get enough oxygen into their blood. So that happens with the type 1 cells. So the type 2 cells don't do the gas exchange. They're too thick. They're too thick. But the alveolar type 2 cell has a very important role, which is to produce this. Here they're calling it a surfactant layer. Here they're calling it a fluid surface. The picture here is better. That these may remind you of lipids. If you took biology, which you all did, I'm hoping this reminds you of lipid molecules. And its purpose is to keep the airway open. If you took away these molecules, if you took away these lipid molecules, the airway collapses. And then you can imagine this top wall is stuck to this bottom wall, and there is no airspace. It's gone. It's collapsed. So it should be when you inhale, airspace opens. And when you exhale, it closes a little bit, but not down to nothing. So my son was born at 33 weeks, my first son. And preemies, premature babies, don't make this. And so their lungs are really, they work very poorly. So he went into intensive care, neonatal intensive care, and they had to administer through an inhaler this surfactant, which allowed his lungs to work. So without it, he would have died. And the take-home message, it's this cell that makes the surfactant. The type 2 cell makes this essential layer, and the type 2 cell is the one that is targeted by the virus because it has the ACE2 receptors. And I think my next slide shows this. Oh, it doesn't. It's the next slide. Let me go to the next slide. And it's these bodies that are the storage place for the surfactant. The surfactant is lipids. These are lipids. This type 2 cell secretes some of this lipid, makes the surfactant layer, allows a human to breathe. So if the virus attacks this cell particularly, knocks out the ability of it to secrete the surfactant. Without surfactant, the airspace collapses. This is what's killing people with this disease. This is showing the actual cells. So here's the real cell, not the cartoon, the real cell. And these are the lamellar bodies, LB, Laguna Beach, lamellar body. There you go. And this is an electron micrograph where you can see they look a little different, right? They look a little different. Instead of being the flat surface, they're these domes. And that's why here they're saying, yeah, there's the flat surface, and there are these domes now and then. So those are the cells that the virus knocks out. So here's a much zoomed out picture. You have lungs. In your lungs, you have type 1 cells and type 2 cells. I don't like that they made it look like there's a whole lot of type 2 cells. They're not, it should be the other way around. It should be 1 type 2 cell for every 7 type 1 cells. So maybe they just messed up on their diagram. There's the surfactant layer. This is the healthy lung. And everything works to get oxygen into your blood. These are the blood cells in the capillary that pick up the oxygen as they go through the lungs. And on the right side here, this is, they're saying injured. And these are all the things that happen with this disease. I'm not going to go through all the complicated pictures here, but you have inflammation, you have fluids that collect, that's the pneumonia. You have swelling of the cells that makes it hard to breathe. So this is what's happening in the people who are severely affected by the virus. This will be my last slide before I ask for questions, but this is a cool slide because if viral entry is step one, there are clever humans saying, can we block it? Can we block that step one and prevent the virus from getting in? This low resolution image here is the natural way it's done. If you are infected and you recover, you will have made antibodies naturally that stick to the virus and block it so it can't enter the cell. So this is nature, and it's been working for 40,000 years for Homo sapiens. But if you fail to make the antibodies, you may die. With this virus, a decent probability that you will die. So then humans are saying, can we help out nature? Can we help out nature with science? So the cartoons kind of do a good picture of showing what scientists are thinking of. Could you make something, this little blue oval, that sticks to the ACE2, and then if it's stuck, then the virus can't. This particular idea is to say humans, scientists can make this structure here in a lab and just throw it into the human, and if it's there, then the virus, its docking site is filled. It's a ship coming into dock, and they're saying, there's already a ship there, I have to wait. I can't get to that cell. This one is a different approach. It's not the viral protein, but the effect is the same. Make something that sticks to the ACE2 receptor, and if it's docked, then the virus can't come in. This last strategy here is a different approach. This is to say, can we make decoys? That one's a decoy. Take this ACE2 receptor, make it in a lab, and throw a whole bunch of that into a human, and then the virus will say, I have found my target, but it's wrong. It's a decoy. And since the virus is bound to this structure, it can't get to the real target, which is the cell. So all of these approaches are being tried to block viral entry, and a vaccine, if we get one, would be trying to say, you can develop antibodies before you're infected. That's the vaccine approach. Okay, pause for questions. So once again, I'll monitor the chat, and you can ask me about anything you've seen so far, or maybe things you haven't seen. Chat, 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 chat. Okay, so we've got a question from Bronson Bailey saying, what's the difference between type 2 and type 1? I think you're talking about the type 2 and type 1 cells. They are, I'll go backwards for that. They're really quite different. The only thing they have in common is they're in the same space. They're in the lung here. But as far as what they look like, maybe this is the best. This is a cell. You know, this thing here that you say, what is that flat surface? It's a cell that's just been like flattened, like you rolled out a pancake until it's a tortilla. It's a tortilla. So this is a tortilla cell, and this is an olive. And so they don't look anything alike. They don't have the same functions. They just exist in the same space. And that's why they're called type 1 and type 2. Dolly is asking what causes the swelling. Thalia tarasi. Okay, so all these letters here are, the official name is cytokines. They're signaling molecules. They are used, when it's working right, they are used to signal your body to say, invader is present, let's go attack it. So for example, TNF is tissue necrosis factor. Necrosis is death, saying, let's go attack it, let's go kill him. There are things called natural killer cells that go out and are supposed to protect you. But it can get out of control where it's not only damaging the intruder, it's also damaging the healthy cells. And that's the process that's known as inflammation. You get inflammation sometimes when you say, my finger's swelling up because I got a cut, and you say it's red, it's puffy, it hurts. That's inflammation. And you can imagine that same process occurring in a lung where you're saying it's red, it's puffy, it hurts. That's what's happening here. Oh, okay, Zoe Gort is asking, so you can only get this virus once. Nobody knows for sure. The usual, I was just thinking about this the other day. There's some viruses, definitely, you can only get it once. Once you've got it, you're immune for the rest of your life. Smallpox was one of those. And so there is no smallpox anymore in the world, because it was, you get it once, you're done, and humans made a good vaccine. Measles is also, humans have a good vaccine. Some people, well, never mind. Measles is good. Flu is not. Flu, if you get flu this year, you can get it again next year because the flu virus will have mutated. This coronavirus is probably more like the flu, and so probably will mutate, but nobody knows for sure because we've only had it for 3 months in this world that we live in. So the hope is that you can only get it once, but the reality may be, no, if you got it this year, you could get a different strain next year. All right, Andrew Kaiser asks, what is the role of ACE2 receptors in normal function? Yeah, good question, Andrew. That's because it does have a real function. It's not there so the virus can enter. It's there to do something important. And I don't think I have the time to discuss that in great detail, but this ACE2 here is an enzyme, as it says here, and it's important with this is a signaling molecule, angiotensin. So ACE2, you'll appreciate this, Andrew. ACE2 is a protease, and it cuts angiotensin 2. And so if you block its function, you certainly wouldn't want to block it permanently, because then you as a human will lose this vital function. The short answer is it's involved in blood pressure regulation. So if you block it, then your blood pressure is going to do weird things. But humans have a lot of blood pressure medication, so you probably can try to undo what you did. Yeah, good question though. Chantel Carter has the last question. Are there examples of the therapeutic blocks being used for other viruses, and what would the development process be for creating them? Excellent questions. I'm not going to try to answer that. It's an excellent question. Maybe I can get to it in my next webinar. The short answer is I cannot specifically talk about viruses where this approach has been effective, and I'd rather be specific than general. So that's why I'm not going to try to answer this question. But I will. I thought about this last week too, or maybe it was just yesterday, that the only, other than the vaccine, which has wiped out a lot of terrible human diseases, the only kind of clever approach that's worked for humans that I know of is HIV. So HIV, humans did develop these ways to block entry and to prevent it from replicating once it's inside. And so that's why HIV used to be death sentence. and now is not. The clever humans made it so that virus wouldn't kill people anymore. All right, if you're rich enough to live in America, as my wife said. Okay, let's go forward. Well, too far. Okay, so now we're on stage 2, which is viral replication. So once the virus gets into your cell, it needs to replicate. For that to happen, this is the most important part. The outside is just to get the RNA into the cell. All this outside is just for that purpose. Then once the RNA gets inside the cell, time to replicate. So we're here, we talked about the ACE2, and now here's the RNA. Here's the RNA. And the goal is to make many viruses, many viruses. So this RNA needs to be copied maybe 10,000 times. And you need, with that RNA, you need to make all the outside. You need to make the outside of the virus. So this is a very simple cartoon. I'm hoping you've seen things like this in biology, where this is DNA. Virus has RNA, but the concept is the same. Here's the DNA, and the goal is to end up with 2 copies of it. So the blue DNA is the original, and this kind of pink is the copy. And so you just make, you make a copy, you make a copy, you make a copy, and then you have 2. An enzyme is required to duplicate or replicate either DNA or RNA. So this is an enzyme here, this gray blob. Enzymes are proteins, they do things. I have a wonderful video to show you, I think it's on the next slide, of the polymerase doing its thing, because it's not static. It doesn't just sit here, and it's not slow either. But the bottom strand here is, we'll call it complete, and the top one is being synthesized one at a time. T first, G next, A, C. The next one should be a T. So this enzyme will take this, which is just floating around in the cell, and it will add it right here, and it will make all the bonds that are needed. And then it will go to the next one and say, okay, the next one, if you know your biology, the next one should be G. Next one should be G. You can see by the shape that the G would fit here. So this polymerase, it's going to add a nucleotide, is what it's called. Then it's going to add another one, and another one, and another one. And by the time it gets to the end, it will have made the copy, the duplicate. So the virus needs to do that. Needs to make copies of its RNA. The previous slide had one gray blob to illustrate the enzyme. This more complicated picture shows that for the virus, it's how many proteins is this? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, maybe 10 proteins coming together. And this is a machine. It's amazing. So I'm going to show you this video on my next slide. But let me just make the connections here. This red is the original DNA strand. This blue is the enzyme. This yellow string coming out here, that's the copy. That's the copy. When you're copying RNA, it comes out as a single strand, not a double strand. And then these little yellow things floating around, those are the building blocks that make up the RNA or DNA. They're these things. One at a time, one at a time, one at a time, one at a time. Okay, so the one at a times are here. They get bonded together in a chain here. This enzyme is reading the information from the original strand. Okay, so this will be It has narration, so maybe I'll let the narrator do her job, but I may put some interjections in from time to time because this is wonderful.
Process, reading off the information that will be needed to make the protein.
That's the enzyme.
Everything is ready to roll. 3, 2, 1, go.
And there it goes. That's the
The
enzyme.
molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the 2 strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.
Okay, so that is the polymerase real time. Goes really fast. Let me just start it over a little bit here and pause it when I want to.
Needed to make the protein.
Okay, so that's like the 10 proteins I had in my slide.
Everything is ready to roll. 3, 2, 1, go.
Meow.
The molecule
Oh,
racing along the DNA is
stop that. Sorry.
Everything is ready to roll.
3, 2, 1,
1,
go. And now pause. Perfect. Okay. So it said at the end of that, this is occurring in every cell of your body. True. If you are infected with the virus, you've got additional copying going on, and that's the virus copying its RNA. But you can see it just kind of goes along really fast, and it makes this copy. That's the polymerase. And I think that's all I need for that. Okay. Boom. So the virus replicates really fast. So this is a recent paper in Journal of Virology. They measured 2 types of viral replication in tissue culture. So I'll show you what that means in a little bit. This is not inside a human. This is in a little dish, in a petri dish. But you can see they infected the cells, and then they said, how about 3 hours later, 6, 9, 12 hours later? This TCID is a measure of infectious virus particles. So if you don't want to deal with the complication, just say number of viruses. This is the 1,000. So after 3 hours, they said we can get 1,000 virus particles in each milliliter. After 6 hours, saying it's 100,000. After 12 hours, it's 1 billion. It's 10 to the 9th virus particles in each milliliter. This is half a day, and it's gone from 1,000 to 1 billion virus particles. This, on the other hand, is measuring viral RNA, and it's not quite as dramatic, but it's still it's still amazing. So here we've got 3 hours after infection. This is 10 to the 4th. We're saying, let's not even worry about that. Let's just say what happened in 3 hours, going from T equals 3 to T equals 6 hours. In 3 hours, it increased by a factor of 1,000. So the viral RNA didn't double in 3 hours. It went up by 1,000-fold in 3 hours. So this is also what makes this a frightening virus, is it replicates quickly. But that's typical of viruses. They've got to, as I said earlier, they've got to do their job, replicate quickly, get out before the immune system destroys them, infect the next person. All right, pause for questions here. So you can ask me questions about anything that I presented recently or before, it's fine. Still waiting for my first question. I should plot the questions versus time graph, because I think it does get to the point where their brains get tired. And it's hard to think of questions when your brain gets tired. Going to wait 20 more seconds. And on we go. So if you heard what I said earlier, the data that was presented was from what's called tissue culture. And tissue culture is where you grow cells. These are cells here. You can see their outlines. And you grow them in a dish, so you can do experiments. You can do viral studies on cells growing in a dish. So this is a pretty simple cartoon, but you take your virus stock, which could be, for example, here, they had cells growing in a dish, and they said, let's take a sample of the supernatant, the liquid above the cells, and let's test it for virus. And that's what they did here at 3, 6, 9, 12 hours. So they said, let's take a little bit of the supernatant and let's test it on some new cells and see how much virus is in the supernatant. So you usually do it this way. Sequential dilution is what it's called, because this is probably so much virus that it'll wipe out your entire cell layer. I'll show you a picture on the next slide. So you do dilutions and you keep diluting and you keep diluting and you keep diluting. And then you say, well, how many viruses are in this sample? How many viruses are here? And you can see a kind of a cartoon here saying there are these little white circles that are called plaques, and you count them. You count the plaques. And this is what it looks like in real life. So the purple, that's what's called a lawn of cells. You grow cells in a dish and they grow kind of like the grass in your lawn. So they grow until they touch each other. They completely fill the dish. And wherever you see purple, those are cells. Wherever you see white, that's called a plaque. And that's where a virus infected the cells here and killed them. So white spaces are dead cells or no cells. They were killed by the virus. So you can see it's easily countable. This one might be 2 possibly, but let's just count it as 1. 1, 2, 3, 4, 5 plaques here. And this was a dilution of 10 to the minus 5. So that would mean you have 5 times 10 to the 5th viruses in the original stock solution. And you can see this dilution, there are no purple cells left. There were so many viruses that it wiped out the whole plate. All the cells died. So this is a way you can count live viruses. How many are there? You count them this way. And that's one thing that's clearly very important, to be able to count how many live viruses are there in this sample. That explains how you got this data. Tissue culture infectious dose, how many plaques were there in each milliliter? And they're saying 1 billion. If we used 1 milliliter of this supernatant, it would kill 1 billion cells. So you dilute it until it only kills 5 or 50 cells. The second graph, they were measuring the RNA itself. So if you saw my webinar on PCR, polymerase chain reaction, that's how they do that too. How they quantify quantitate how much virus is with PCR. And if you didn't see that, then I'm not going to try to explain it, but it's a machine, and you put the right things in, and it tells you the amount of viral RNA that's in your sample. And it usually takes a couple hours to do. And so that's how they got this data, is they put the samples into polymerase chain reaction PCR and said, okay, well, there's 1,000 times more RNA at t equals 6 hours than there was at t equals 3 hours. Boom. All right, this is how I get to end. This has been, let me say that more politically correct. This is a drug. It's made by Gilead, which is a California company. They're in the Bay Area. And their whole job is to make new medicines. So this is the name of the drug. This is the structure of the molecule. And there have been trials to test this. Does it help this disease? Does this drug help cure the disease? And just 2 days ago, this is Anthony Fauci, who's the government's top scientist, let's say it that way. And he says, there is proof the drug can block the coronavirus. It is not blocking entry, it is blocking replication. So this next slide is an attempt to explain, how does it do that? So here's the polymerase that's supposed to chug along really fast, right? Really fast. It's not really chugging, it's zipping. And it's supposed to add one of these bases at a time. Ka-chink, 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 and you saw it goes really, really fast. This is remdesivir, RDV, remdesivir. It looks like ATP. And ATP, you have to imagine this is an A up here. Instead of a T, imagine this is an A. That's a building block. So the remdesivir is supposed to look enough like this molecule here that the polymerase will take it in. But as it gets taken in, which if you remember the video, these little things are flying in, things flying in, flying in, flying in, and they're used to make the growing RNA chain. Remdesivir gets taken in, but it jams the mechanism. So the polymerase stops. So if you can imagine sticking this in here, and instead of going to the next one, it says, ugh, stuck. It's like a lawnmower hit a rock that got stuck, and the lawnmower is therefore stuck. So this is a very clever approach. This is the way a lot of drugs are made now, as you say, there's a reason to believe it should work. I will make it. I will test it. And if it does work, then Gilead makes millions of dollars and stockholders are happy. And if you can afford it, you're happy too. So that's a little political commentary on top of the scientific commentary. But this so far is the only drug that has scientific data that looks really reliable. The other drugs that have been sort of, try this, try that, there's not scientific data saying it works. So this could be a big deal here, that if you find something that works, then it does give ideas for the next round and say, because this didn't work very well. I should clarify. Yeah, it worked. It made it so people were sick for severely ill for 11 days instead of 15 on average, which is good. It's helping. But still 10% of the people who got this drug died. So it's not a miracle cure, but perhaps it is the first step to saying, okay, well, if that works a little bit, maybe this will work better, or maybe it's a combination. That's what happened with HIV. It was a combination of a molecule like this that blocked the polymerase, and then another molecule that blocked another part of the viral life cycle, and another drug that blocked that part. And when you took all three in combination, the virus was no longer killing people. So that's the end of my show. I can take some questions. So I'll once again look at the chat, or if you want to raise your hand and ask a question out loud, we can do that too. Looking at the chat. Oh, I can talk about Chantel's question a little bit too. Chantel's question, the second part of it was what would the development process be for creating therapeutic molecules that can block viruses? So in, I think it's 4 weeks, the 4th week, we'll get to have a webinar from Andrew Raub, that's his job, is to make the molecules that can cure cancer. So he's a PhD chemist, ACR class of 2007. He was the first class, I think. So if you can tune into that one, you'll learn about the drug discovery process. Okay, Luke asked the question, does remdesivir interfere with DNA replication of regular human cells? Clearly very important. If you're saying this blocks the virus polymerase, you don't want it to block the human polymerase. So it doesn't. That's why it can be used as a medicine. If it blocked the human polymerase, it's a poison. It just kills people straight up. But since it specifically blocks the virus polymerase, then it can be used as a medicine. And it was originally developed for Ebola virus. Didn't work on Ebola, but now it may work on this coronavirus. Okay, I'm going to wait a few more seconds. The questions have been excellent, so thank you all who participated. You helped your peers learn more, and you helped me make a better presentation.
So there's a question from Adam.
From Adam in the chat? Ah, there it is. I missed it. Sorry. Great. From Adam T. Is there any kind of regulation to prevent the treatments from being unreasonably expensive? So the American way is the drugs you can charge, you patent them, right? You patent your drug. So Gilead has the patent for remdesivir. And then you say, then it's up to Gilead to set the pricing. So the first things I read about Gilead, is that they were saying, we will not try to bleed you dry for this medicine, which is great if true. But the United States way is, if you invented it, then you should profit from that invention. So other countries are less like that. I mean, and the United States has always been make $1 billion, invent something, make $1 billion. Who cares about the public? So we'll see with this. We'll see with this, and it may lead to changes, right? There are a lot of people in this country who are saying the way we do things is wrong. It's not humane. It is inhumane to say only those who can afford get the medicine. Okay, so that's probably a good way to end today's webinar. Thank you all for being here. There will be, there may not be, let me do this. It says here until next time. It may be that next week I don't do a virus webinar. There's certainly plenty more I can talk about, but the scheduling next week may not allow me to do the webinar in the usual way. But there will be another one in 2 weeks for sure. And I'm learning a lot. Hope you are too. Time to say goodbye for today.
Bye. Thank you.
Thank you, Mr. Sogo.
Thank you.
Thank you.